Hello and welcome back to our series on genetic regulation. In this lecture, we are going to discuss one of the most ubiquitous or widely used mechanisms of genetic control, transcriptional regulation via transcription factors. This is a very important lecture because, as mentioned, transcriptional control of genes via the use of transcription factors is how nearly every gene in eukaryotes is regulated. For the most part, transcription in eukaryotes does not take place without transcription factors. So listen up. In this lecture, we are going to be discussing the use of transcription factors to regulate transcription. Recall that transcription is the process of reading DNA into pre-mRNA, or mRNA that has yet to be processed, which is splicing, capping, and polyadenylating. Essentially, we're going to be discussing how we can get RNA polymerase to do its job. A quick reminder that genes can be classified in 3 ways. Constitutive genes are always expressed, meaning that they are always transcribed and translated. Inducible genes are those that are normally off but can be turned on. Repressible genes are those that are on but can be turned off. In the case of transcription factors, when we talk about a gene being on or off, we are talking about whether RNA polymerase can transcribe the gene into mRNA or not. In the case of constitutive genes, RNA polymerase can easily access and transcribe a gene into mRNA. Recall that this is the case for many genes that are responsible for the production of proteins that are essential for life, such as ATP synthase. Inducible genes are normally off, meaning that RNA polymerase is not always transcribing that gene. Although it may be bound to the gene's promoter, it's not actively transcribing the gene. In order for it to transcribe the gene, it has to be told that it can do so. Essentially, in inducible genes, RNA polymerase is sitting at a stoplight, waiting for it to turn green. Repressible genes are the opposite. RNA polymerase binds to the promoter of these genes and actively transcribes them. However, if necessary, this process can be stopped. Essentially, in repressible genes, RNA polymerase is cruising down the road through green lights, but it can stop if it reaches a stoplight. Using the same analogy, in constitutive genes, RNA polymerase is cruising down the freeway, meaning it is not regulated by traffic lights. So for inducible and repressible genes, which are regulated by molecular traffic lights, what are the traffic lights? Well, they are DNA binding proteins called transcription factors. Before we understand transcription factors, we need to revisit the structure of a gene and actually understand it in a little more depth. Recall that a promoter is where RNA polymerase binds to a gene in order to initiate transcription. Essentially, the promoter is the start for transcription. The promoter is actually a little more detailed than simply a place where RNA binds. In fact, it's composed of 2 main parts. The core promoter, shown here, composed of the transcriptional start site, and, well, which is where RNA polymerase binds, and a Tata box, which is a region of DNA composed of T's and A's, thus the name, where transcription factors bind. Upstream, or in a region of DNA that comes before the core promoter, is another important region of the promoter, the regulatory region. This region varies depending on whether the gene is inducible or repressible. If a gene is inducible, this region will contain an enhancer region. In this case, transcription factors called activators will bind and will work together with the transcription factors bound to the Tata box to induce or activate transcription. Essentially, if a gene has an enhancer region, RNA polymerase is sitting at a red traffic light, but binding of activators to the enhancer region will turn the traffic light green and enable transcription to progress. If a gene is repressible, this region will contain a silencer region. In this case, transcription factors called repressors will bind and will work together with the transcription factors bound to the TATA box to repress or stop transcription. Essentially, if a gene has a silencer region, RNA polymerase is cruising through a green light, but binding of repressors to the silencer region will turn the traffic light red and stop transcription from occurring. Sometimes, promoters will contain both enhancers and silencers, enabling the genes to be told to stop or go anytime. I attempted to clarify the structure of a promoter and an entire gene a little bit in this image. An incredible work of graphic design, if I may say so myself. Recall that a gene starts at a promoter and ends at a terminator. Transcription of the gene into mRNA therefore starts at the promoter site and progresses until it reaches the terminal region or the terminator. This process reads the coding region of DNA into pre-mRNA, which is then processed into mRNA, which can then be translated into protein. As mentioned on the previous slide, the promoter or the start site of a gene is a little more complex than just an area where RNA polymerase binds. It is composed of a core promoter, which is composed of the transcriptional start site, where RNA polymerase binds, and a TATA box, where transcription factors bind. Within the promoter region, but upstream of or before the core promoter, is where the regulatory region is. This is where our enhancer or silencer regions are located. Essentially, RNA polymerase and some transcription factors can bind to the core promoter, but unless activators bind to the enhancer region, transcription will not progress. Or RNA polymerase might bind to the core promoter and be told by repressors that it cannot progress. So the enhancer or silencer regions serve as a molecular traffic light, controlling whether RNA polymerase can transcribe a gene or not. Okay, to better understand the structure of a gene, more specifically, in order to better understand how transcription factors regulate transcription, we need to understand how they interact with both RNA polymerase and DNA. Although prior to this lecture, we've discussed transcription as progressing due to the binding of RNA polymerase to the promoter, this is not really the full story. RNA polymerase actually binds to the transcriptional start site, but not alone. It binds in a multi-protein complex called the pre-initiation complex. This is a complex of proteins, including RNA polymerase, which is shown in purple, plus a bunch of other proteins, which are called transcription factors. Transcription factors are often called TFs, and this is what you're seeing in gold in this image. As you can see, some transcription factors bind directly to RNA polymerase, while others bind to the TATA box. But all of them, collectively, as the pre-initiation complex, are binding to the core promoter. Remember, the core promoter is composed of the TATA box plus the transcriptional start site. Notice this transcription factor on the TATA box. This transcription factor basically holds RNA in place until it gets a signal from the enhancer or silencer region to either go or stop, respectively. If this transcription factor gets the signal from the enhancer region to go in the form of an activator, this transcription factor will kind of just like let go of RNA polymerase, and RNA polymerase will be free to transcribe the gene. If this transcription factor gets a signal from the silencer region to stop in the form of a repressor, this transcription factor will basically just like hold RNA polymerase in place, and it won't be able to progress and transcribe the gene. Let's explore this more closely by exploring each of these options individually. Just to orient you to the structure of a gene again, you have the core promoter composed of the TATA box and the transcriptional binding site, the coding region of a gene, which ends in the terminator. Upstream of all this, you have the regulatory region, which is composed of either a silencer or an enhancer or both. RNA polymerase binds, I think I have to do this. Let me get there, sorry. I made pretty animations. Let me get it. Right. RNA polymerase binds to the transcriptional start site, and transcription factors bind to the TATA box and to RNA polymerase to collectively form the pre-initiation complex. In an inducible gene, hold on just a second. In an inducible gene, a transcription factor called an activator would then bind to the enhancer region. Recall that binding of the activator to the enhancer region turns on transcription by activating the pre-initiation complex and enabling RNA polymerase to read this DNA into mRNA. But how it does this is actually pretty incredible. What happens after the activator binds to the enhancer is that this DNA actually bends to enable the activator to physically activate the pre-initiation complex. Boom! This bending is facilitated by a DNA bending protein. Once the activator binds to the pre-initiation complex, RNA polymerase is activated, and thus it is able to read the coding region of the gene into RNA. There we go. Repressors work similarly, sort of. You have your core promoter composed of the TATA box and a transcriptional start site, the coding regions of your gene or the nucleotides that you need to actively transcribe into mRNA, and you have the end of the gene, the terminator. Upstream of your gene, you have the regulatory region. RNA polymerase binds to the transcriptional start site. Transcription factors bind to RNA polymerase and the TATA box to form the pre-initiation complex. In this form, RNA polymerase is essentially waiting on directives to tell it whether it can go or not. When a repressor binds to a silencer, the DNA is bent, and the association of the repressor with the pre-initiation complex essentially holds RNA polymerase in place, preventing it from transcribing the gene into mRNA. So to summarize this process, the structure of a gene is composed of the gene, the segment of DNA that is to be read into mRNA during transcription. This segment runs between the promoter, or the beginning of the gene, and the terminator, which is the end of the gene. The promoter is composed of the core promoter, which contains the transcriptional start site, and the TATA box, and the upstream regulatory region, which contains either an enhancer region or a silencer region, or both. Transcription factors and RNA polymerase bind to the core promoter to form the pre-initiation complex. In this state, RNA polymerase is essentially held in place until it receives the directives to stop or go. If an activator binds to the enhancer region, RNA polymerase will be released to transcribe a gene into mRNA. If a repressor binds to the silencer region, RNA polymerase will essentially be held in place and transcription will not be able to progress. Oops. Hold on just a second. I actually have a really good video that explains the entire process of transcription and translation. But even better, it has a visual of how transcription is activated with the use of transcription factors. So I do want to share this video with you. I'm going to see if I can get it to work. So if you give me a second here. Sorry. See if I can get it to work. Yes.
DNA's most extraordinary.
Okay.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood.
This is your pre-initiation complex.
This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process.
That is your pre-initiation complex. So that's RNA polymerase plus some transcription factors located at the core promoter.
Reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3.
What you're about to see, right? So this is a gene that's under inducible control. You have the pre-initiation complex bound to the core promoter, and what is attached to the enhancer region is an activator. So what you're going to see is this pre-initiation complex be activated, or this gene to be induced or turned on. So remember, your pre-initiation complex sits on the core promoter, and your activator comes around, bends the DNA, binds to this complex, which activates and basically releases RNA polymerase to transcribe the gene.
2, 1.
Let's watch that again. Okay.
Everything's ready to roll. 3, 2, 1.
Amazing. So that was the activator binding to the pre-initiation complex and allowing RNA polymerase to be released to transcribe the gene. I'm not going to watch the rest of the video just due to time, but I did link it here. Definitely watch that video. It's really excellent. This is also a really good video I wanted to show you. So let's watch it.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TA-TA sequence in the promoter.
Basal factors are another term for transcription factors. So transcription factors and basal factors are the same thing.
Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers scattered around the chromosome can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
All right. So to tie this all together, I want to remind you that cells often respond to stimuli with genetic change. Previously, we mentioned how this genetic change is often facilitated by genetic regulation. Now we can specify how this takes place. As you can see here, when a cell receives a stimulus or a signal, it triggers signal transduction, cell communication, or cell signaling. And this leads to a response. That response can come in many forms, but often it is in the form of a transcription factor. These are usually activators or repressors, either inducing or inhibiting or stopping the transcription of certain genes. For example, if the signaling molecule was a pathogen, such as a bacteria, this would lead to transcription factors activating genes involved in the immune response. Or if the signal was from a nearby cell, this could stimulate transcription factors inhibiting the expression of genes that stimulate cell division. Transcription factors are one of the most ubiquitously used mechanisms for enabling a cell to respond to its environment. From this lecture, make sure that you understand the terms constitutive genes, inducible genes, repressible genes, core promoter, Tata box, enhancer region, silencer region, transcription factors, activators, repressors, and pre-initiation complex. Make sure that you understand the structure of a regulated gene and how genes can be turned on or off via the use of transcription factors, activators, or repressors. And understand why transcription factors are a common effector response resulting from cell communication. Thank you so much for watching this lecture. I'll see you next time.